All right, here. So, welcome back, everyone, to the Blue Rose webinars, the translational webinars. And we are talking tonight about the fundamentals of genetics for clinicians. Because in the past, we quite often get from the doctors that they didn't really do genetics in their undergrad, and some of these terms can be sort of overwhelming. And once again, tonight, I'm lucky enough to not be alone. I'm joined by Professor Stephen Jeffery. Stephen, how are you?
Oh, good. Thanks, Chris. Wonderful. It's nice and hot here in Cape Town today. But I'm very excited by the topic of the discussion today. And yeah, so I'm really quite keen to see how you can help us and how you can actually get us on board as clinicians.
Yeah, absolutely. I was thinking tonight what we can talk about is sort of the basics of how inheritance actually moves through families, what the DNA report is doing when you see the word pathogenic variant, um, what we can now actually sequence with panels and exomes and that type of thing, and why gene editing and ethics and all of these things are suddenly a conversation that you hear even in the clinic. So by the end, you should be able to answer questions like, how can this run in my family, and what tests can I use, and what do they actually measure, and what do the results mean once I look at the genetics biologically, and of course, what's just hype and what's actual genetics. So I think, as usually as these things go, we should start out with a little bit of history. Um, in terms of how we came to the place where we can now look at genetic variants and choose which ones to look at, in terms of deciding which disease risks are there and which profiles people have and what medications to prescribe them. So somewhere in the mid 1800s, a guy called Gregor Mendel, was at a church there, was breeding pea plants. And what he noticed is that some of them have purple flowers and some of them have white flowers. And he could, when he selectively bred them, he could actually predict what the next generation will be. And so that was the start of it all, this sort of predicting of inheritance patterns. Also in the 1800s, we eventually discovered chromosomes and the process of meiosis. And so this was the foundation of genes becoming an idea. But the actual concept of DNA as the genetic material actually only got accepted in popular science in the mid 1900s. And in fact, in the mid 19 in the 1950s, actually, three very intelligent people, namely Watson, Crick, and Rosalind Franklin, discovered the structure of DNA. And since then, there's just been a cascade of from recombinant DNA to sequencing to genomics, the Human Genome Project, and modern genetic tests. So there's really been a massive cascade. And I see we're joined by a guest tonight. Lovely. How are you tonight?
Hi, I'm fine, thank you. Apologies for being a little bit late.
No problem. No problem. Uh, so yeah, Doctor, would you like to introduce yourself as well?
Let me, sorry,
Yeah, yeah,
I can introduce Dr. Brina. So Dr. Brina is a
brilliant.
um, women's health specialist, number one, but has a broad experience in large range of clinical medicine, ranging from everything from acute care to general medicine, and really brings with her a wealth of clinical um, background to these discussions. And so she's just going to join with us as well and give us an idea about some of the more broader aspects of what patients might need, and especially female patients, actually, which is the large part of what she sees on a day-to-day basis. Um, so I think it's just great to bring that clinical perspective into today's discussions. So yeah,
Absolutely.
so Carmen,
Absolutely, yes.
Chris has really just been giving us a historical background, and for those of us who did medicine, just the Mendelian stuff and the more basic stuff doesn't bring back the best memories of medical school. Genetics wasn't something that we were taught well. We often fail to see the real, true clinical application. And certainly what we have access to today wasn't really available then. So, yeah, so I mean, it's great. And I think Chris is really was just really getting us started on a not only the history, but really just trying to understand, where we've started and where genetics has began through to really seeing how those applications can come in today. And so I think it's going to be great. In common, please feel free to interrupt and take the discussion where you'd like it to go as well. So thanks so much.
Yeah, absolutely. Please interrupt anytime. I'm looking forward to hearing a diverse set of eyes and set of opinions and these type of things. So yeah, I was just talking about the the quick rate at which genetics has developed and how we these days we're already at the point where you have gene therapies and genetic editing and CRISPR was a buzzword not that long ago, and even that's all technology now. There's a new one called Tiger TAS, which has replaced not replaced, but is a sort of cousin to CRISPR and the gene editing space these days.
What is the difference?
Yeah, so, yeah, Karen, sorry.
Yeah, yeah.
I
Well,
just kind of like maybe you can, in a easier terms, simpler terms, explain what the difference is, because I always get a bit confused with CRISPR.
yeah, well, CRISPR I know well. Unfortunately, I haven't had the privilege to really work with Tiger yet, so I don't know that much about its molecular mechanics. But CRISPR is quite a simple process. So CRISPR stands for clustered regularly interspersed short palindromic repeats, and it's little sections in the DNA that repeat. And because of this, you can use something called the guide RNA to go find these sequences. Then the Cas9 protein, or the Cas13 or Cas12, depending on which type of CRISPR system you're using, goes to that specific location and then it cuts the DNA. Once a cut's been performed, you can, if you have gene insertions, you can actually give it material which will then either fill that space, or if there's no other material, that space will just kind of close itself back up again and fix itself. So that's the basic principle of CRISPR. It finds these sort of repeated sections and then makes a cut there. In fact, that's the basis of most genetic editing. Even before CRISPR, we had things like zinc finger nucleases and talins and meganucleases, and all that any of those really are. It's some enzyme that finds a specific location and makes a cut in the DNA. So I'm assuming Tiger will probably work on the same mechanism, but I don't know. I haven't actually looked at the molecular mechanisms for Tiger yet.
Okay.
Yeah.
And then what's the like examples of the application of this in healthcare?
Yeah, so there's quite a few. In fact, quite recently, I think about a month ago now, there was a case where they treated a type of, I believe it was deafness. I always get confused. It might have been blindness, but I think it was a type of deafness that they did. That was just a simple single gene mutation that they had to treat. And what they did is they did the treatment with a gene therapy approach using CRISPR, and it's showing promising results. And so we actually at a point now where in human applications, they are applying these sort of genetic editing technologies to prevent even chronic and lifelong debilitating disorders. So unfortunately, I don't remember the exact story, whether it was deafness or blindness and which type of deafness or blindness it was, but the the concept of them using this technology is the same.
Okay.
Yeah. Yeah. So and I'm very, very optimistic for the future where there's so many diseases that are what's called monogenic or just have single variants involved with their expression. And so like sickle cell anemia, for instance, is a single little letter difference that causes red blood cells to be misshaped, essentially.
Yeah.
And this is something that could theoretically be fixed. So yeah, I'm very optimistic for when we get to a point where we're able to, in a whole body, sort of give this gene therapy, because that is ultimately where the issue lies, is to take a single cell and edit that. It's not too difficult, right? You can change the nucleus and the DNA in that. The problem comes with, okay, but how do you then take that mutated cell and spread it to the whole rest of the body? Last week,
Yes, yeah, that would be the next step, right? I mean, how
yeah,
do you?
absolutely. So
Because we
there's all sorts. Yeah,
yeah.
exactly. Yeah. No, exactly. So
Because you
yeah,
use so I worked a little bit in research actually for quite a few years, and I was familiar with it. They did a lot of sort of genetic stuff when they were looking at viruses, and we particularly the SARS-CoV-2 virus, where they were looking at looking at the different genetic mutations, and every time like the different variants. So from that application, for vaccine development, that's a great tool. But when you're looking at diseases where there's like multiple, like millions and millions of nuclei compared to a bacterium, it's a bit, yeah.
Absolutely. Yeah, absolutely. And it's so last week we spoke a little bit about pharmacogenomics, and we spoke about these concepts of pharmacokinetics and dynamics, which in a nutshell is either what does the drug do to the body, or once the drug is in the body, how does it spread? Um, and uh, and that is the problem, is do you use a viral vector or a bacterial vector, and how do you actually get this to apply to the entire body? And like you said, to use it to maybe find a specific antibody in the case of a vaccine or something like that. That's a lot easier when you're working with a single thing. But when you're working with these complex systems where there's literally millions of possible interactions, it becomes. But apparently that's what Tiger, this new one, is really good at, is spreading. So maybe that'll be a topic for a future webinar, the ins and outs of this Tiger system. But we'll need to get well, I'm not an expert on that, so we'll need to get an expert on that to come explain that mechanism to me as well. Yeah.
So, Chris, do you think that, I mean, obviously part of this is people who really got diseases, like you said sickle cell and many of the other genetic diseases, but, the huge application also being preventative medicine, which is in many cases the holy grail of medicine, trying to identify people at early age, diabetes, cardiac, dementia, even cancer, and trying to see how you can modify the expression of those genes in the body. And that's obviously people are working on that, but does anyone know how close we are to finding some sort of therapy that, I mean, I know you're talking about Tiger and things like that, but how does it look one day? I mean, it's, that's really medicine at a completely new level that we never had seen.
Yeah. You see that the problem with the preventative, when we talk about gene editing, so last week we spoke a little bit about the difference between somatic mutation and germline mutation, right? So just the recap of that, that's when, in the case of something like a cancer, is if a cell gets a mutation and then it causes this sort of rapid growth, and that's localized in the cell. So if it's a germline mutation, that's something that's inherited to the next generation. Similarly, when we talk about editing, so you can have somatic editing and you can have germline editing. Now, if you're talking about a preventative way, so let's say we take one individual and we do genetic editing that prevents all future individuals from this person to have a disease, then you're talking about germline editing. And there's a lot of ethics surrounding doing this sort of editing, where you're editing something that can be inherited. And a large argument for it is that we don't necessarily know, we aren't 100% sure what the risks are or can be, especially 10 generations down the line. And also it's the whole ethical consideration of consent. The potential future generations never consented to having their genomes edited. There's a famous movie called Gattaca, where the entire civilization is based on these sort of people who are selected based on their genome for what job they can do in society and that type of thing. And so, yeah, when we're talking about preventative medicine, that's one thing, to prevent disorders and diseases, but it is also, people often say, a slippery slope to the argument of, have you heard of the concept of designer babies, for instance?
Absolutely, yeah.
Yeah. So that's always the risk, and it's it's a conversation that is always ongoing, and rightfully so. But it is also one that we don't seem to come to a conclusion with, and it is also one that makes it difficult to employ some of these preventative methods of gene editing, which is why we mostly looking at it from a secondary or a primary point of care perspective, where someone is born with a disease and we try and see if we can cure it in that individual. But then the transport of the medication, in this case, the genome editing, becomes a very difficult issue. Or yeah, it's a complex problem. It's not complicated necessarily, but it's a complex problem to solve. So on that, so let's talk a little bit about, I've spoken a lot about hereditary and sort of somatic, but let's just take a step back and think, but okay, how, when you speak about something being inherited, what is it that's actually being passed down to the next family? Because if you if you think about the colloquial definition of inheritance, right? That's when a family member passes away and you get their furniture or their house or that type of thing. Now, genes and genetic material obviously doesn't work like that. If someone has diabetes, that doesn't mean that you get for sure diabetes. You have these sort of packages, the DNA packages, and these are called alleles. Now, allele is the half of the gene that you get from the one parent, and you get the other allele from the other parent. And these two alleles together make what's called your genotype, right? So, or the gene that you have. And this is done, or this then is used to predict what your phenotype would be. So you have your genotype, which is your two alleles, one of which you got from each parent, and this relates to your phenotype. The phenotype is the clinical expression. So that's the patient coming into your practice, and they have this thing expressed. And from that, you can infer, oh, okay, so you potentially have this genetic combination because I can see the phenotype with my own eyes. And if you really want to go into the sort of molecular biology of that, so an allele is technically, so you have the difference between mitosis and meiosis. Mitosis is any cell division. So you get a wound and the cells divide and the skin grows back. Meiosis is a two-step process, and it results ultimately in gametes. So that's egg cells and sperm cells that are haploid. So that means they only have one copy of each chromosome, right? The reason why that's important, why cells are haploid as opposed to diploid, is because if they were diploid, uh, then we would have a doubling with each generation. So the next we have 23 pairs of chromosomes. The next would have 46. The next would have 80, or 92. So it would, and obviously those would not be viable. So that's why we have this, and that's why from the haploid organism is where you get your allele. Just to sort of zoom into the actual molecular side of things. But when it comes to the actual clinical, the patient sitting in front of you, there's very few terms that are really relevant. So off the top of my head, I can think of dominant, autosomal dominant. So you can see if a person has one allele. So in the case of high impact genes like the BRCA gene and the FH genes, Lynch genes, you can see if the person has inherited one of these alleles from one of their parents, that means they're at risk. In the case of BRCA for breast cancer, you get autosomal recessive type of genes. So usually that means they're only at risk if they have, sort of to put it in simple terms, if they get the bad allele from both of the different parents, and then you have the X-linked one. So that's something like color blindness, for instance, which is linked to the X chromosome. That's also why you would see something like males being much more likely to be colorblind, because they have one X chromosome and one Y chromosome. So if they have the mutation for a color blindness on the X chromosome, that's the only X chromosome, so they definitely have color blindness. Whereas if a female who has two X chromosomes has a mutation on one of them, she still has one working X chromosome, and then it's not expressed as color blindness. There's actually another important way that these things get inherited, and that's through extranuclear DNA. So that's the DNA that you find in mitochondria, right? And that's actually passed on only via the mother. So I wonder if I may ask, in the sort of realm of obstetrics and gynecology, do you ever sort of deal with a lot of these maternally inherited diseases? Is that a topic that comes up ever in the field?
Um, not actually. These aren't large and huge conversations, that we do talk about. Specifically relating to prenatal testing, Chris, or is that what you're talking about in terms of what, how you would then analyze whether the person is going to continue with the pregnancy. Is that what you're talking about? Or are you talking more about the patient themselves being diagnosed with the disease?
Well, it's more the diagnosis.
Yeah, yeah.
Yeah, because there are some phenotypes that can be carried in the mitochondrial DNA. So that's what happens there essentially is if the mother has a large percentage of affected mitochondrial DNA, then it's very likely that they'll pass it on to their children. So that's not something that gets screened for, and it's not really in discussion at all when it comes to um, gynecology and this type of thing. People don't really look at mitochondrial. It's a bit of a trick question. I know they don't actually test for it, and uh, yeah, it's a strange thing that they don't test for it because it's one of those things where if you see a percentage of, let's say, mutated mitochondrial DNA, you can almost directly predict what the chances are that the children will also be affected by the same extranuclear derived diseases. And yet there's no screening for it and there's no conversation about it.
So just
So
remind
it's
me, Chris, sorry, so what are the mitochondrial associated diseases then? I know you didn't
the
mention a few. Which ones are they?
yeah, so there's it's one of those cases where you can't actually say that this disease is directly connected to mitochondria, um, statistically, right? So there are always going to be other factors, but there's, for instance, the a famous phenotype. So it's or rather phenotypes and diseases, and a famous phenotype is this sort of fatigued phenotype. So it's a you get examples of people that are fatigued. They have a tiredness, and that's directly linked to the mitochondrial expression, right? Because I'm sure everyone probably remembers one little fact about mitochondria from high school, which is it's the powerhouse of the cell. It's where the ATP comes from. All right. Yeah. So yeah, I see, Stephen, I see you asking something, but you're muted.
Oh yeah, no. It's I agree. I mean, people did agree with that. That is probably the one thing we do remember. But I mean, I think from my recent reading, this is maybe slightly separate. Is that when it comes to your ability to, on the opposite scale, your fitness and your muscle memory, I think that is often associated with mitochondria. And do you think fitness and sport capability is more transferred through the mum's genes than through the dad's genes? I'm just suddenly thinking a completely new part of the subject, maybe a little bit
Yeah, no, I
not
like
to
the
quickly talk
question.
about while it's still top of my mind.
Yeah,
What
I
are your
love the
thoughts?
question. Yeah, it's great. No, it's absolutely there would be a direct link to it because if you're getting all of your mitochondria where the Krebs cycle happens, where all the ATP is made from the mother, then your sort of natural fitness and Krebs cycle and carbon cycle machines are all coming from the mother. So yes, that is connected. And I don't think there isn't a lot of studies on fitness and sports expression and how that links to mitochondrial inheritance from the mother and ATP levels. So that's a very interesting study actually that would be worth looking at. We just need to find a clinical application for it.
Well, I mean, I just remember very clearly reading a lot around sport and fitness and our ability to stay healthy into our old age, and very, very clearly, it seems that even someone who might have been very fit previously, that you retain your muscle fitness in a way by virtue of the fact that you trained your mitochondria. So
Yeah,
it's
but
very much mitochondrial based what you are doing when you're doing your exercise and doing fitness. And so
yeah.
I'm just really randomly in a very sort of novice way looking at it and wondering if there is some sort of association there. But I suppose it is a little bit off topic. But
Well,
it might be quite interesting to look at that.
that's the nature of DNA and genetics is because it's the foundation of all biology. Nothing is off topic, right? Because it's mitochondria and these things are, yeah, it's always going to be relevant. And so, yes, but just to answer your question there on the fitness thing is, yes, it is the mitochondria that is directly, it's ATP. And even when you look at health supplements like creatine, for instance, what that essentially does is it all comes back down to the Krebs cycle, the ATP synthesis and all of that happens in the mitochondria. So yes, it's a very interesting link.
Yeah, well, you've tied that in very interestingly because, I did a very quick AI search for supplements. I'm sorry, there's some noise in the background. It's kids. But I just recently just did an AI search on the best supplements, because, if you look at what people are taking, and I mean, Carmen can definitely come in on this because, she's having these discussions every day with patients. But, what do you do to promote longevity? And it's creatine came up very strongly. We're taking omega-3s and 6s, and I've got patients who are taking a handful of things. But
Yeah,
I mean, but creatine really seems to be something that came out. Maybe
absolutely.
comment your opinion on what you're suggesting to patients. What are the supplements that people should be taking for long-term health and longevity, etc. Mute.
Carmen, you just muted.
Sorry. Yeah, so I really don't suggest heaps of supplements for my patients, to be honest. I'm very much more dietary focused. And if patients are of a certain age, then perhaps they might require some replacement. But creatine is an interesting one because there's been a lot of talk about it because there was a study that was released related to looking at the risk between, a reduced incidence of Alzheimer's and creatine. So there's this big focus on sort of the neurologic cognitive function and creatine. So a lot of women who are entering sort of the midlife, the perimenopause stage, they're now all having this brain fog and they're all now overdosing on creatine. But there's no clear evidence yet that of the benefits with that. I'm not too sure really of the physiology, how the creatine would improve that based on its sort of impact on the Krebs cycle or on that, on the mitochondria. I'm not too sure about that. But creatine is just a flavor of the month at the moment. Before it was collagen, and collagen is also something that a lot of people for skin, for anti-aging and wrinkles, and yeah, so I think those are the two big ones. But I mean, the list of supplements is just there's like NAD is another one that patients, they go for IV drips to get NAD, which apparently and I don't know that much about NAD, but it works at a DNA level, I believe.
Yeah,
That's how they market it. I'm not entirely sure. Does it really
I can
work?
comment.
Because, yes,
I
I
can
would
comment on
love
that.
to hear about NAD because
So
there's NAD in IV form and then there's NAD also in oral form that patients are taking now. And then there's also a injectable that is on the market from the
yeah,
compounding pharmacies. So
yeah, and it's not to alienate any future sponsors, but NAD plus or NAD, the evidence behind it isn't very strong. So there's another one called nicotinamide mononucleotide, which is kind of the chemically the precursor to what would eventually become NADH
yes,
and NA.
NMN. Yes,
Yeah,
yes,
NMN.
yes,
And
yes.
that works a little bit better in terms of the
In an
pharmaco.
oral form, because I
Yeah, and
always
then
wonder, you're taking all these things and it's going to the gut, or the stomach, which has got a high acidity. How do you know what at the end of the day, what is, being absorbed?
yeah, absolutely. And what your particular gut microbiome does with it as
Exactly, because
well.
that also will metabolize it.
Yeah,
So
Yeah,
we
that's the
incorporate it
problem.
into its own genome. So it's yeah.
yeah,
How
absolutely.
does
Yeah.
it, so how does that work then, the NMN?
So yes, it does eventually also end up in the mitochondria and the whole Krebs cycle thing. And it just, yeah, ultimately, Stephen asked about the longevity. So in terms of longevity supplements, that seems to be one that preliminary has some decent links to cellular energy. And similarly to the study that you mentioned about the Alzheimer's, I don't know if it was the same study, but I saw a similar study where they were taking, I think it was 25 grams of creatine daily, and how this showed that there is some cognitive benefit, and all that it really comes down to is cellularly, there's more energy. like
Okay.
ATP synthesis. But it
Okay.
is also in that study, it does also show that that's all it shows, that there is more energy, but whether or not that can be translated directly to a reduction in Alzheimer's, it's at best the correlation. We can't really claim causation with that type of thing. Where we can sort of start claiming causation is with a 5 gram creatine daily for muscle tissue power exertion and that type of thing. So sports science wise, that seems to have a fairly strong evidence base. Now, when it comes to the neurological side of things, yeah, we can't claim it as a miracle supplement. And there was another buzz supplement a while back, lion's mane, and everyone when
Yeah.
it came,
We're
yeah,
talking about
mushroom,
a lot. My patients talk about all the time.
yeah,
Uh,
it's
Never
yeah,
heard
yeah,
of it.
yeah,
Weird,
yeah,
whatever that is.
And
yeah,
how does that also work at a cellular level in the mitochondria as well? I mean, because that's also for energy and for clarity, for focus. Because there's quite a few mushrooms that are like all the rage,
yeah,
reishi, lion's mane, and there's another
yeah.
one. But
Yeah, so well, none of those have very very strong significance when it comes to the actual correlation tests. So
mm,
at best we can see some sort of trends, but yeah, no, it doesn't have the same sort of clinical significance as say paracetamol would have on on pain or whatever. So it's not a if it were at the level where we would claim direct sort of administration of this leads to a betterment, we would, of course, pharmaceutical companies would be selling it in bulk. So, but creatine does have a lot of good things
Look, I
going
mean, I
for
think
it. Yeah, yeah,
look, I
yeah,
think I mean, I love this discussion because it's actually really taking gene therapy to a level where we are actually attempting gene therapy. I'm not saying we're doing it, but and again, I think what Carmen brought up was very interesting. I mean, I didn't think about the IV administration of some of this stuff, but we're probably not getting the right dosing. We're not getting the right administration in, and we're going to have to, the pharmaceutical companies and the industry will have to think of novel ways of administering these things so that, we do have some drugs where they've got certain capsules. Like we've got a long-acting anticholinergic that we use, and it's in a capsule form that only gets absorbed deep in the colon or way down the gut. So, trying to avoid the first part metabolism because you don't actually know when that lion's mane is taken, what does it actually do? It gets into the liver, it gets metabolized. Does it end up in the same way? I mean, you spoke about collagen. I never really thought collagen was a good idea. I say to my patients, rather just spend that money on a nice steak, you'll feel better. It's it's protein. I don't really know if those amino acids or whatever they are actually going to end up going to your wrinkles in your face at all. I feel that it was a bit of a waste of money for a lot of those patients. So, but again, it's also like the probiotics. Where do we start with that? It's a completely different discussion, not really genes, but what type. What happens when you take them. We know we're onto something here, but I think there's so much research that needs to happen on this level.
yeah.
Um, I mean, do you think there's a way of doing a lab-based assessment? Because to can't try and get 2 million patients to take lion's mane and see if they live till 85 rather than 83. I mean, I don't know, that's impossible. But can we somehow take this into a lab and replicate this in the lab? I mean, AI is there. Can we model the stuff? Those are all the things that I think we need to ask ourselves.
Yeah, yeah, and we're heading towards that direction. So when you're talking about in silico analysis, that's exactly it. When we kind of use a computer and we use machine learning or AI to look at the many genomes so entire sets of genes from people that we have available, and then we are able to look at pr predictions, right? Correlations, whether this is correlated to another drug. There's an entire database of these sort of gene to drug correlations. It's called PharmGKB. And it's not that there's ClinVar, there's quite a few databases that link certain genetic variants or genes or pathways to certain drugs or physiological outcomes and that type of thing. And there's a lot and speaking of genomes, where do you find this 2 million people or 8 million people, whatever it may be, there's even a lot of consortiums that do that type of thing. the first big one was called the Human Genome Project, right? And then, yeah, but since then there's been I think it's called the Africa 100 or H100 for Africa Genome Project. So there's been a sort of African-centered genome collection, and there's been a European-centered genome collection, and there's all sorts of these sort of databases. And we in the age where we now have all of this information, and we have statistical models that we can use to make correlation and best cases. And then you asked, okay, but then where do we go from here that it's actually a clinical application? And before we get from correlation to human, there's always the slightly uncomfortable question, but the way that science is done a lot of times is with animal models, right? Or bacterial models or model organisms. And so, yeah, and that's I think what also last week you asked me, why is there a rate limiting step, or why does it take a while for this sort of clinical trial process to get from correlation to drug, or something that you can say this genome equals plus that drug equals this result? And it's because, well, somewhere along the line we need to test this and other things before we can test it in humans, and the ethics around that is can be uncomfortable. What I look forward to is a future where we can skip that whole process, and just do it all on a computer. That where we have these sort of synthetic computerized simulations that you can see this gene, it works like this. We know exactly how it works. If you give it this drug, we know with an exact percentage that this is the amount of brain growth that you'll get, or energy from the mitochondria that you'll get, or something like that, based on your profile. Now, we're not there yet. We're very far from there, and whether we'll ever be there is questionable, but not impossible.
Agree with you. I was actually talking to Carmen yesterday about the some of the work that Kit Parker's doing in Boston and Harvard, and his main lab is focused on modeling of physiology. We were there a few years ago, and in his lab itself, he's replicated the blood-brain barrier, he's
Oh, wow.
replicating the placental barrier, and able to do research on that. He's created islet cells for pancreas therapy in his lab too. So without doubt, people are working on a physiological interface or physiological modeling.
Yeah.
And it might be interesting to have some sort of biological hybrid system with an electronic attached. So these are just ideas for you clever guys, Chris, to look at building something like this. But the sky's the limit in a way, and electronic stuff, chips, they're getting very cheap, and maybe we could ultimately have something that you guys can build in your lab. When you board one afternoon in your so that's yes.
Yeah, I love our little R&D afternoons where we didn't have an idea for a product in the morning, and by the afternoon we have a a brand new.
Yeah.
Last time you walked in there, we were doing a augmentation biology sensory pack. Remember?
Yeah,
The
no, it was, yeah. So as I say, I think we don't know what technology will enable us to do, but
yeah,
those days of giving, we were talking about drug studies as well yesterday, and I think those days are hopefully going to be over soon, because it's tedious research
yeah,
and it's messy. You look at some of the biggest epidemiological studies that have ever been done, they remain difficult to interpret. As an
yeah,
example, the Women's Health Initiative, which was a menopausal study, the Nurses' Health Study. I get a headache when I start seeing people talk about these trials, because although they brought in thousands of women, still very hard to know what to say, and it's because of
yeah,
that genetic variation, that multifactorial aspect. It's
yeah,
not
yeah.
it's not being
Complexity.
accounted for, and they want to, and so we're at the mercy of statisticians to interpret messy data that's multifactorial, and so and a drug company is desperate to get some sort of meaningful results to make their investment, make sense. And so, and I think that's really been one of the big problems of modern medicine, and
Yeah.
and the answer will absolutely lie in genetics, because that's what it will explain the variations in patients, between
Yeah.
different studies, etc.
Yeah, well, I'm biased in saying it, in agreeing with that the answer lies in genetics, but yeah, or at the very least, the sort of interdisciplinary connection between the scientists and the clinicians and the pharmaceutical companies and all of these places. And not to open a can of worms, but I think academia has a little bit of a blame with these sort of difficult to interpret statistical papers as well, because there is a very high incentive to publish a lot. And so we were talking about it the other day, where something that would have been a PhD project 20 years ago, was a master's project 10 years ago, was an honors project 5 years ago, is a undergraduate task this year, right? With the rate that things improve the whole time. And so, for instance, with a single variant, it used to be a case that you could publish a whole paper on one single letter in the DNA that was different, a single SNP, single nucleotide polymorphism. These days, a single paper has thousands of these variants, and with machine learning, millions of these sequences that you can code. And so it is an interesting thing but it's data, it's messy, and as you say, you're at the mercy of bioinformaticians and statisticians to look at this and interpret it. And yeah, and even they, because that's the problem. It's like you go to a statistician and they don't know the biology. You go to a biologist and they struggle with the stats and don't know where the medical application is. You go to the doctor and they don't know the biological underpinnings. So it's this lack
Well,
of communication between the fields.
we all know that what creates difficult to interpret data is the heterogeneity of things, and I think we have not included the very important genetic variations between patients in studies, and that's maybe explained some of the more very difficult to interpret studies. And I think that's, I hope we can actually include that at some stage. I mean, Con, what are your thoughts on this whole issue of messy data and messy studies?
Yeah, I think it's just become such a competitive field, research, and more so in the last couple of years, when the focus really was a lot on research since the pandemic. And now it's like everyone needs to come out with the latest and the newest, and we need to look for this. And they really are trying to make things more and more difficult. I mean, I saw it firsthand working in a research unit where our projects were becoming more and more impossible to do and to analyze. We had to, because you have to compete with what that one's doing. That one released another paper in Chile and this one in Australia and that one that. So we must do better than that. And it's become quite a competitive industry, really. And I feel a lot of the times we're not really answering any questions. We're just creating more and more. And that's really not what research is supposed to be about. I think we just I feel like there's just more and more gaps, and we're supposed to be, filling the gaps in research. But
I agree.
I just feel it's gone the wrong way, personally.
Absolutely. I think as a surgeon, I was involved in some surgical trials, and I was highly critical of a very simple thing like the randomized control in surgery, because, they would recruit and randomize patients, like maybe 55 patients in each arm. It's very difficult to interpret any results from that, and I think we have run away from the important aspect of really collaborating in research. I think, academics often have the wrong attitude, because we don't work together. People want to compete with each other, and it's driven by metrics that are bizarre. Like, everyone wants to count the number of publications that a certain person has, and they could be publishing studies that don't really make sense to anybody, but it's, that's the driver. Whereas you've brought it up immediately, whereas if we took, we all work, and I think the need for consortia is much more important. If people are doing cholesterol research, maybe they should be putting their data into much bigger databases that can be crunched quickly by the machine learning and the AI thing. So, I think we really do need to have a complete mind shift on how research is done. Sorry for the noisings. No one's being murdered. It's another kid. Sorry.
Yeah, I completely agree with that as well. And as you say, we are incentivized in research a lot of time it's almost a bit of a dog eat dog world. And you want your department to outcompete the other department, and you want your impact score to be higher in your department. And it's just, you've pinned the people against each other that really ought to be working
Yeah.
together.
Yeah. And it's like if you had a rugby team when everyone was trying to like work out who was going to get the most ball or score the most tries or make the most distance. I mean, it would never work. But I mean, it's only put everyone together. We might actually get there. But to try and change that is impossible. And I don't see it happening in my lifetime. I mean, if you look at the competitiveness, even locally, it's quite insane. And then obviously you get the big discrepancy between investigator-initiated research, and then you've got the pharmaceutical research, how research gets funded. And admittedly, people need to make a living, and how you do that is also driven by what you do and how your own performance is judged. So I think it's made it very difficult. But I believe that new ways of looking at research and data, like genetics, I think will change things a lot, and I look forward to that. And maybe the new generation of researchers will see it like that. And I think maybe in the next 10 to 15 years we'll see a new mindset when the generation that is now growing up in the world of AI, and I'm sorry to use the word AI because for want of not having a better terminology, but maybe that generation will see things completely differently, and maybe people like Carmen's daughter who will look at the world a little bit more critically and not be driven by the same things that we are driven by, because maybe things will be easier, or there might be other metrics that we're able to generate an income on, and we can put our minds to research in a better way. So yeah, but I mean that's a little bit more philosophical discussion.
Yeah, but it's a hopeful one, and I do share that sort of hope that the next generation will be looking at more interdisciplinary and more result and betterment incentivized studies, and we do have like you said as well, there is a financial incentive around a lot of these studies, and yes, people need to make a living, but there's a big difference between people making a living and people exploiting others to make millions.
Yeah.
So
that's a whole other
yeah.
discussion.
Yeah. No, but
Especially in Africa, yes.
yeah, yeah, absolutely. But yeah, I do personally, I have a lot of hope. And once again, I'm biased as a geneticist, but I have a lot of hope with the information that we get from this sort of blueprint that is DNA and RNA as well. If you just look at all the applications of it, and and the how knowing the very basic science has led to really phenomenal increases in what we can do. You mentioned the SARS COVID vaccines that were made. Now, of course, there's always a little bit of politics, and probably a lot of it bleeds into the discussion that we just had with the financial incentives and all of that. But taking the good out of that, we did really quickly come up with a really effective way of administering a vaccine based on messenger RNA, which is a very clever thing. Because then, so to take it back to some to some fundamentals of genetics, in genetics, there's this concept of, they call it the central dogma. Now, it's a great starting point when you're learning genetics, but as you sort of progress further, it becomes less and less relevant, and there's more and more exceptions to it. But the central dogma essentially says you start out with DNA, which is kind of like the the architect making the rules of what needs to be coded. Then from there, the DNA makes RNA or messenger RNA, which is kind of like the blueprint or the instructions. And then that goes to a cell or part of a cell called a ribosome. And the RNA, messenger RNA binds there, and there's a whole transcription thing that happens, and eventually you're left with different proteins, enzymes, receptors,
Remember the
transporters.
ribosomes, because those were the other little dots in the cells that we learned at university.
Yeah. They are the other little dots.
We know
Yeah.
what they look like. They're little round dots in the cell.
Yeah.
They went in the middle of
Yeah, exactly. And yeah, and so I think the SARS-CoV vaccine is brilliant because what it essentially did, it said, well, what if we just give the cells the blueprint? What if we just give it messenger RNA? And what we found is, yeah, the ribosomes, the little factories, they take it and they make it. They just made the spike protein. That's brilliant. And it's using such a fundamental basic concept and making a whole new medicine out of it, really. Well, a vaccine's not a new medicine. It's a very old concept, but still, it's actually, I don't want to use the word miraculous because that's not what it is, but it's phenomenal. It's impressive that what we can do with just the basics of science and genetics,
Chris, do you
and
think
putting our minds together.
so? Do you think that happened because there was a worldwide global problem and there was just a completely different incentive to get something sorted from the COVID vaccine? Everyone knew that there was a problem, and there was a much bigger incentive to find something quickly so that everyone could get back to normal life. And so there was a more collaborative effort. People let their guard down and work together, or was that not really the apparent driver, do you think?
Yeah, well, I mean, I can give my opinion. I think so. I think that sort of having a unified front against and a different incentive to do this type of thing did create a sort of shift in focus. Of course, I can't claim with certainty that that's why we
Mm.
were able to do that, but as a
Makes sense.
layperson,
That people know.
yeah, seems that
Makes
way
sense
to me.
that there was a different incentive to
Yeah,
get it fixed quickly, and it would be nice if the other world's problems could be fixed in that way. The
yeah,
other huge genetic things. Because you
yeah,
think, I
yeah,
mean, it took them like 9 months to come up with a vaccine, and there are so many other world problems that they can't fix.
yeah,
Like
exactly. Yeah,
like and you wonder, diabetes, it's been around for decades, but they still, and you start wondering about incentives and de-incentives to get these things fixed, But
yeah.
I think that's something
It's
for different discussion.
it is, and I know a very interesting. I actually know a few professors there, and people in the labs there at Tigerberg who work on rare genetics, rare disease genetics, and that would actually be a real because. So the reason I say this is because a lot of times these studies are which study you do and which study gets funding is incentivized by what has immediate sort of danger. So this is a disease that we focus on because people are directly dying from it, um, type of thing. But that lab, what they do is they specifically look at the rare diseases and they specifically look at the things that are, for instance, chronic and not immediately life-threatening, but have disability adjusted life years that extend for an entire lifetime. And yeah, so that is also, I think it's also just a human way of viewing things. This is if something is going to kill you now, you want to solve it now. But if something going to kill you in 30 years or just make the next 80 years uncomfortable, I think there is maybe less of a urgency to try and solve it. But that's just speculation. I don't know. You know, the
Anyway, that's another interesting topic, and just looking at, as I said, some of the biggest diseases in the world, and how little progress has been made. You'll probably live longer with your cardiovascular disease, but actually modification and risk reduction and those things very difficult to do. Alzheimer's, a lot of the cancers, people still die from those things, and statistically we haven't made as much progress as we think.
yeah,
And I read somewhere that the longevity that we see today over the last 100 years in the world is in large part related to our overcoming infectious diseases and good antibiotic use. But
yep,
people still die from very similar things to what they were dying from 50 to 100 years ago.
yep,
And I mean, that to me is very disappointing, and
yeah, and that,
where we are failing.
Yeah, no, I agree. I agree 100%. And that's exactly what I'm referring to actually is we had a massive incentive to look at something like infectious diseases and sort that out quickly. And now we're stuck with the stuff that is more complex inherently, but yeah, but it did have this sort of long-term thing, but it has become the disease or the disorder of our age because that is now what we're mostly dealing with actually and mostly dying from. Um, and yeah, no, it's
Yeah,
a
I think oncology is a big place where this can really work because pharmaceuticals are not, you look at the chronic diseases, right? And they don't really want there to be a cure, do they? I mean, hypertension, they don't really I must be honest, do they really?
Well, I
Big
mean,
pharma.
that's the point I've been trying to make in the last few minutes, Carmen. And you just said it quite straight. But I mean, that's what I always say. I'm sure they found it. They
Yeah,
have a cure for diabetes somewhere. They have a cure for HIV. They have a cure for hypertension somewhere, but they don't want to give it to us because
exactly.
you can
But
imagine
for
the amount of
cancer,
money that will go.
okay, we can cure
Yeah.
cancer because there's no, the treatments are very expensive. Very few people can actually afford them. So there's no big market for the chemo drugs as such. So we can cure cancer, but those chronic diseases are a problem. Um, but that's where
Well,
we are needed. But then we can look at other things like obesity as a disease now. It's classified as a disease, and I think that's actually a very important disease, which is the root of a lot of these lifestyle diseases, and that's a disease that needs to be focused on because it's polygenic. I mean, there's lots of genetic
you're right.
mutations that could possibly happen that you could target treatment for.
yeah, yeah, absolutely. That and
I mean,
the metabolic syndrome. So diabetes
yeah.
is an example of that. And yeah, no, I don't know. I don't know if there are these sort of. It sounds a little bit conspiratorial if you say that with a lot of the chronic, but it's true.
Well,
That's yeah.
well, a lot of money gets pumped into various hypertensive medications and all those drug trials, isn't it?
Yeah,
Yeah, I
yeah,
mean, and that's actually, to be frank, why we struggle with antibiotic resistance, and the pipeline for antibiotics is very short because they're not chronic medications.
yeah,
If you're sick, you'll take an antibiotic for maybe maximum a few weeks, and you'll either get better or not. So there's no it does often come down to incentives.
yeah,
Yeah,
yeah.
yeah.
I agree.
Sure, we are running out of time, Chris.
We
I think it's
are,
been a
we
great
are.
conversation. I think we've opened a lot of can of worms. We might have offended a few people. We might have people after us, so we missed out for a few days. But I think we touched on a lot of very important aspects of modern medicine, and yeah, we look forward to a few further discussion on these points. I mean, I want to thank Carmen for joining. I think her contributions and the insights were really great. And yeah,
Thank you.
what are we talking about next week, Chris?
Well, I had a different topic in mind, but with all of the talk of CRISPR and Tiger and gene editing and gene therapy, I'm wondering if we should talk about uh, that topic, gene therapy.
I'd love
So
to hear that. I'd still
yeah,
don't understand how you can take the genes and therapy them. It doesn't make sense to me because your genes are your genes. You know what I mean? But I'm looking for.
yeah,
Yeah,
yeah. So let's make that the topic of next week. Yeah, and thank you very much, Stephen and Carmen, for joining. It was absolutely amazing to hear your opinions on these things as well, and just help get the insights into the more clinical side of things, because yeah, from the science side, you don't see patients, so you don't know what's going on on that side of
Mm.
things. So yeah, thank you. I really appreciate your inputs.
Thank you.
thanks, Chris.
Thanks so
Thanks,
much.
Carmen. Okay,
Okay.
bye-bye.
All right, cheerio.
Bye. Bye.
Bye.